Hi, and welcome back to The Sound of Science. I'm your host, Amy Myers, and today we are going to be talking about more of the properties of biological molecules. Specifically, today we're going to focus on the AP Biology sections 1.5 and 1.6. This covers nucleic acids, and of course, those are our DNA and RNA. And so we're going to get into a little bit more detail about the DNA and RNA as we discuss nucleic acids. DNA is structured in such a way that the orientation of the pentose sugar, that 5-carbon sugar, matters. So if you take a look at our diagram here of our carbohydrate, here is an oxygen. And then to the right of the oxygen, we have carbon 1, carbon 2, 3, 4, and then up here on a stick, essentially, is carbon 5. So carbon 5, or 5 prime, is where our phosphate group is going to attach. Our 3 prime carbon is where we're going to have a hydroxyl group attached. And so in that regard, when we talk about carbons being either a 5 prime to 3 prime orientation or a 3 prime to 5 prime orientation, that's what we're getting at. We are getting at the carbon, which numbered carbon each item is attached to. So when we look at the double-stranded DNA, the 5 prime to 3 prime on one side pairs across to a 3 prime to 5 prime on the other. This means that the strands run anti-parallel to one another. So when we start 5 prime here and run to 3 prime, on the other side, we'll start at a 3 prime carbon and we'll run to a 5 prime. And as you can see, the pentose sugar is actually, it looks like it's upside down from our perspective because of that. In the center at the nitrogenous bases, we know that thymine and adenine will pair together. And because of the structure of those molecules, adenine and thymine can actually form 2 hydrogen bonds between them. Guanine and cytosine, again, have a different structure, and they are going to form 3 hydrogen bonds together. Hydrogen bonds are going to help stabilize the molecule by allowing the 2 sides of the double-stranded DNA to attach together, but those bonds can also be broken fairly easily to allow for replication and transcription. Let's talk about how we actually build this strand. And we're going to cover this again a little bit later when we talk about protein synthesis and we talk about genetics, but right now, the things that I want you to kind of recall from your freshman or sophomore year biology class is that nucleotides can only be added at the 3 prime end of the growing nucleic acid. So in this case, with the strand moving, we have essentially what leads to a leading and a lagging strand. So moving in one direction, these can all attach because we're moving that 3 prime to, or that 5 prime to 3 prime direction. So these are attaching to the 3 prime, and they can do it in a straight line. When we talk about the other side, however, we actually have to build them a little bit at a time, little segments. The nucleotides or the nucleic acids are going to form together. They're going to bond together using covalent bonds where those electrons get shared. This is what's going to allow us to make the strand. So here we are comparing DNA and RNA. And this is really that 1.6 segment. We're just going to sneak it in here in the middle of 1.5. So DNA and RNA both have nucleotides that have that 5-carbon sugar. In addition, they both have that phosphate group and they both have nitrogenous bases. Each nucleotide is going to be connected by covalent bonds, and the covalent bonds are actually what allows us to create that sugar phosphate backbone. Linear strands are going to have that 5 prime and 3 prime end. Okay, so all of that makes sense. Now let's talk about how they're different. So DNA is a deoxyribonucleic acid. Deoxyribose is a sugar that is missing an oxygen. So if you take a look here on this diagram, you can see that we have just a standard hydrogen here. Whereas if we look at our ribonucleic acid on the other side under RNA, it has an oxygen attached. So this is a deoxygenated ribose, deoxygenated 5-carbon sugar. This one has the oxygen there, so it's considered RNA because it isn't missing the oxygen, right? Super simple. DNA is able to utilize the nitrogenous base thymine. Thymine, which we normally represent with a T, usually pairs with adenine. In the RNA molecule, because of this change in structure to this ribose, the change in that formula, thymine can't be used there. So in RNA, we replace thymine with uracil. Uracil will still bond with adenine. We just won't find any thymine in the RNA, only uracil. DNA, again, because of that missing oxygen, is able to be double-stranded. RNA is going to be a single strand. And DNA, because of the double strands, of course, is going to run anti-parallel. And so a lot of this information is repeat information from segment 1.5, which is why I decided to go ahead and handle it here. So if you're following along with closed notes that you've been provided, you might need to skip around a little bit as we move through the remainder of the section. All right, take yourself back to that previous biology class where you talked about how DNA works. So we know that the order of nucleotides is going to determine the order of amino acids, which ultimately, what we've already discussed with proteins, is that the amino acids and the fact that some are hydrophobic, some are hydrophilic, and some are ionic means that the protein's going to fold in a very specific way. If you change the order of amino acids, that folded shape is going to change. So changes to our nucleotide sequence, our structure, will change the protein structure and therefore can change the function of the protein. And as just a quick reminder, remember that when we have our DNA, it's going to pair with the mRNA or pair into mRNA. We'll make a copy into mRNA, which can leave the nucleus. And then that's going to determine which amino acids get placed. So what I have here is an example of an original amino acid sequence and a mutated sequence. And really the only change here, if you look, was here. And because of that change, it actually changes all of the ones that come after it. So it looks like we had a deletion here. And all of the amino acids after are going to be different. This is going to result in a pretty big protein shift or protein change, which will affect the function of that protein. So now let's shift back into talking a little bit more about protein structure. And this is back in the 1.5 segment of the AP information. Proteins are made of a linear chain of amino acids. One end has an amino terminus, the other has the carboxyl terminus. And we've talked about this in previous videos. The amino acids connect to the growing chain at the carboxyl terminus using covalent bonds. So you're going to have your amino acid and its friend's going to come along, another amino acid, and it's going to attach to it using a covalent bond, and it's going to attach where that carboxyl terminus is. That's the direction of growth. Okay. We've talked about the structure of proteins before, how you have your primary structure, your string of beads of amino acids, if you will. Secondary structure, it folds into either twisted alpha helix or into beta sheets that fold together. Then you take that and that becomes your tertiary structure. Your quaternary structure is when we put multiple of those peptides together, and that's going to ultimately make your globular protein, whose shape determines function. Now let's talk a little bit about carbohydrates and directionality. So The direction of the carbohydrate parts can change the overall structure and the function of the molecule. So, for instance, we have alpha and beta glucose. And if you take a peek at the alpha glucose monomers are set up in such a way that the oxygen on one end is actually pointing in a different direction. It's a flip between an oxygen and a hydroxide. So they flip. And that flip actually changes the function of that monomer. So for instance, starch is a 1 to 4 linkage of alpha glucose monomers, whereas cellulose, pretty different from starch, is made up of beta glucose monomers. And it's the same formula. They're just a slightly different structure because of that rotation. That can also affect how it appears. So by having your your carbohydrates, the differences between having alpha or beta glucose molecules, it actually leads us to a different overall structure of the larger molecules. So, for instance, starches like glycogen, which stores energy in humans, has this branching pattern, whereas cellulose is very neatly laid out. This is what we see in cell walls. So, in humans and animals, that branching effect, whereas in plants, we have the same formula but a different structure, it's going to line up and be more linear. This is a huge change in function, and because of that change in structure. Glycogen and starch store energy, but cellulose is going to provide that structure to the plants, different function. So let's do a quick review of what we learned in this video. DNA has a 3' hydroxyl and a 5' phosphate on its sugar. Nucleotides can only be added to the 3' end, and they're going to be added with covalent bonds. Adenine and thymine pair, cytosine and guanine pair, and they are bonded with hydrogen bonds between the two strands that holds the anti-parallel sides together. If we change the nucleotide sequence, that's going to cause changes in the biological information or can cause the stability of the molecule to be compromised. Amino acids have directionality as well. The amino terminus and a carboxyl terminus, amino acids are only added to the carboxyl end of the chain, also with covalent bonds. 4 levels of protein structure: primary, which is your peptide chain; secondary, where you form the alpha helices and beta sheets; tertiary and quaternary. And tertiary, of course, is that one complete. Quaternary is when you have multiple stuck together. Changing the direction of a component in a carbohydrate can change the structure of the complex carbohydrate and its function. So the giant takeaway from this video is directionality matters, structure matters. I hope this has all made sense for you and hasn't been too bad or too dry. As always, I look forward to seeing you next time on The Sound of Science.